Hello, everyone. Christina and I will be co-chairing today's session. My name is Jun Gao, working for Health Canada. The focus of today's session is an update on new or revised guidelines and the proposed guidance from working groups on topics like accelerated approvals and Bayesian statistics for CMC, and also new approach for bioassay performance evaluation and the forward-looking approach for vaccine manufacturing. Speakers from regulatory agencies and the industry will offer a diverse set of insights and will engage in panel discussion as well. Audience are welcome to send questions in chat for each speech at the Q&A session. We are honored to have Matt Verling as our first speaker. Matt holds a position as a senior expert at Medical Products Agency in Sweden, working with quality assessment of human and veterinary biologics and normative work within this field with a focus on vaccines and monoclonal antibodies. A pharmacist by training, Matt has been employed at the agency since 1988. Since 1996, Matt has been heavily involved in writing and in implementing many guidelines, including ICH and the EMA guidelines, as well as EMA industry workshop on setting specifications, making use of prior knowledge in regulatory submissions and the quality expectations in relation to accelerated approvals. Today, Matt's speech is about establishing specifications and the shelf life. Please help me welcome Matt Verling.
So I hope can you see this?
We cannot have not seen your slide yet.
You haven't. That's a pity.
Yeah,
then
no rush.
I'll do it again. Then now I think.
Yes.
Yeah, yes. Start
Indeed,
your starter.
And I will
sharing.
in 2 seconds be in presenting mode as well.
Okay. Yeah, we saw the slides. Yes.
Yeah.
Okay. Perfect.
Do you see them now?
Yes.
Yeah. Perfect.
Excellent. Thank you very much, and thank you very much for inviting me to have this speak. And I must admit, I was quite relieved when I heard some of the early speakers confessing that they were not statisticians, because I'm not either. And but I will give you a little bit of a background on, in particular, on stability aspects and setting specifications for medicinal products, and where we are and what we do see for the future. So what I will talk about would be the first, as I mentioned, a bit of an update on the stability aspects. We have the ICH Q5C, and there is a work starting on that. I will go into that, and then also on setting the specification and how to set acceptance criteria, which is now in ICH Q6A and B. And I think that this disclaimer is quite important in that sense. This is really my position and not necessarily those of MPA or the EMA. So for the stability, that today Q5C is still the global standard, and what is saying is primary data to support a requested storage period for either drug substance or drug product should always be based on long-term, real-time, real condition stability studies. And there's no option for alternatives in the current wording of the guideline. We have Q 1A for small molecules has a wording like alternative approaches can be used when they are scientifically justifiable reasons, but the word alternative appears twice in Q5C and not in the same context. So if you read it very strictly, you have to have real-time, real condition data. This is published in 1996. And is there a need for update? Yes, it is, clearly. And work has started, just started with this, and I think they are actually on their way very soon to Korea for the first face-to-face meeting in that group. So the intention is to merge Q1 and Q5C to a joint guideline covering stability aspect both for small and large molecules. And with doing that, the intention is to include Q8 to Q12 principles, and we will see Q13, 14 when they come as well, but with on criticality, control strategy as a whole, how to monitor the quality of the product. And the update will also be linked to the update of Q6, which I will come to in a while. So what is the chance of getting acceptance for alternatives until this this new version of the guideline is released? It depends on the justification, and I think it shouldn't be taken for granted. But we always have to realize that real-time, real condition will be the comparator. So if there are a risk of not meeting end of shelf life specification, that is unlikely to be accepted. But it is, if you have a good reason and you have some good thoughts on how to justify your and how to justify the extrapolation of stability data to get a longer than shown stability or shelf life, that's absolutely doable. But please take this into account. This will be the sort of the gold standard, at least for now. We do have some shelf life setting may differ where you have the prime products, which is the European, which is in US would be the breakthrough therapy products, where you have products, accelerated products for unmet clinical need. And I will come back to that a little bit later as well. So what are the alternatives? That the models can help, but they should be appropriately justified. And there is a robust, of course, a robust basis is needed to verify the predictability of the model. And you could say a very practical aspect is that in case you assign a shelf life for a drug substance and it appears that this doesn't really, it didn't really work, you would need a shorter shelf life. At least in those cases, all the materials is in your own hands. But if you have a drug product and that is out out in the public, and then it would be much more difficult. So it's just an aspect of consideration that if you haven't got a model, and I think we have seen a reliance on accelerated data to support extrapolation to real conditions shelf life. And it is, I think we have seen it. We have definitely seen it for when you have changes to an already approved one. It's unlikely to allow a significant extrapolation, giving like 24 months date shelf life based on 6 months accelerated data. And I think a wider global discussion between the regulators and industry is needed when these alternatives can be used and what needs to be submitted. So the companies are recommended to seek scientific advice or and in Europe discuss with we have the innovation task force, which is already up and running, and we have what is called the quality implementation or the quality innovation group, which is a new EMA group dealing with modeling and other sort of novel aspects on quality, and go talk with these and have some really early feedback on your plans, because that will help both you and us. So what do we see in regulatory dossiers which are for where you're getting away from real-time, real conditions? So variation, if you have a manufacturing process, drug product, for example, if you have shown comparability between the pre and post-changed accelerated data, the same shelf life in real condition as pre-variation can be applied post-variation. We are expected that real-time real-time studies are run to support this, but it is you don't need to have, you could submit already with 3 or maybe 6 3-month data, I would say, accelerated, and 3 or 6 months data of real condition data could be doable. And then action has to be taken unless if this is not supporting what you thought you would see. And for flu vaccines, we do have a new composition every year due to strain change. So shelf life is applied based on earlier composition, and this may also apply to some COVID products where you use a particular platform. We have seen in the past some predictive stability models combining kinetic modeling and statistical analysis of stability data used to support limited real-time data. I think we had some very few batches, and then this model could support it to support the claimed shelf life. And for the toolbox document, I should explain what it is. It is a document in EU guideline on different tools to use in accelerated procedures, and where you have had an accelerated development, and we are bringing up aspects on control strategy and validation and stability and so on. And in that one, we have the stability described, generated on data from stability of structurally similar molecules. And currently it is limited, I would say, to prime breakthrough therapies or other products for unmet clinical need, but I guess this may be up for discussion as well. It's still an extrapolation, but it's based on justification that data from other products will mirror the future stability of the product in question. And it works best for, I would say, where you have a platform, monoclonal antibodies, for example, that if you have some significantly different products, it's unlikely to fit. But the establishment of the model should be described, and with an understanding which molecules will fit and which will not. And even if you have a monoclonal antibody, some of them will work and some of them won't. And what is important is the actual levels, or sorry, the trend is, the actual level is not important, that what happens, but the trend is that how much does this decrease over time, for example. But then you have to put it in relation to what are your limits and what should be your end of shelf life requirement. And it is not only that it could be that, okay, I got something for a vial stability data, and I want to show this for a pre-filled syringe, for example. you can justify if that is representative, then that can be acceptable. So now going over to the specifications and setting of acceptance criteria, and Q6B is the old workhorse. It's adopted in 1999. It includes well-characterized proteins and polypeptides for recombinant and non-recombinant cell culture expression systems. Principle outline do also apply to other products, certain other proteins, polypeptides from tissues and body fluids. It doesn't cover antibiotics, synthetic peptides, polypeptides, heparins, vaccines, allergens, etc., etc. So I think it is a little bit reflecting on what was the understanding at that time. It's still a good guideline, and I would say at least in most cases, the general principle applies, and it acknowledged that this specification is only part of the overall control strategy and should work together with the in-process controls with GMP, raw material testing, and that IPCs may allow certain attributes to be left out in drug substance and drug product testing. Considerable part of that is on characterization, covering what attributes should be considered for setting the specification. And we have seen a definition, if I remember well, this defined this, what is a product-related substance, what is a product-related impurity, sorry for the typos here, and this definition has been widely accepted and seen, I think, in all applications. There's also a section on attributes to be captured in specification and in different methods used to be used in characterization. And a very small part, less than 1 page, is on how to justify the specification, including the acceptance criteria. And what is then the importance of an updated Q6B? That this is, as you all know, a global business with worldwide activities and contacts with multiple regulatory agencies. And there is it's of key importance that we have a harmonization of the understanding of how to select attributes to be included and how to justify the absence of others, and on what principle should the acceptance criteria be set. The guideline is more than 20 years old. A lot has happened. And again, we have Q8 to Q12, where we have much on process and product understanding, criticality, risk assessment, real-time release testing, quality system, etc. We have a lot of new analytical techniques developed both for monitoring during production and routing batch testing. We have some incredible development of biotech molecules, a lot of monoclonals, other recombinant proteins. We have antibody drug conjugates, we have ATMPs, and a considerably better understanding of product characteristics. But there are some confusion in this guideline and a split message. So these are citations from the guidelines, and it is that it points both to it should be based on data obtained from lots used in preclinical and clinical studies, data from lots used for demonstration of manufacturing consistency, and data from stability studies and relevant development data. And this comes out, but there is also one which is explicitly says that specification should be based on data obtained for lots used in preclinical and clinical studies. Quality of the material made at commercial scale should be representative of the lots used in preclinical and clinical studies. And this is an understatement that these are not always possible to combine, because if we should if we should both look at consistency and clinical justification, whatever we mean with that, we will have a problem that normally you have, and maybe I would say, in contrast to what the situation was 1999, today we have high expressing systems and we have few batches normally used in clinical studies, and their variability may not be sufficient to assign acceptance criteria which will be acceptable for routine testing without the risk of having multiple future out of specs and rejections. And on the other hand, setting specification based on statistical calculation of routine batch results will not by itself guarantee that the levels can be considered clinically meaningful. And a common issues where we have identified in the application is, first one is combined attribute testing. So you have a specification for dimers and aggregates by size exclusion, for example. And we know that dimers are rarely a problem, aggregates may be, and less than 3% may mean 3% of aggregates, even if the standard split is 2.6 dimers and 0.4 aggregates. So it's better to split in separate criteria. We also will have a lot of new testing opportunities with deep sequencing, LC-MS. We will find much more than we ever detected with traditional methods. And how to much larger amount of molecular species versus few by current methodology, that what is the relevance of these peaks? Have they always been there but not been detected? What is the criticality of those? So I think the standard advice is make sure to keep your reference samples to be able to go back to early material when new tests are introduced. If we go further, we do often see making use of statistical tools such as tolerance in limits with very few observations leading to wide acceptance criteria, more like 5-6 standard deviation without the boundaries, far from what has been exposed to patients. And we do also see that you have there is an addition of analytical variability, even if this is already included in the batch data. Adding an extra safety margin due to the limited number of batches tested, and not a single word on how the limits can be considered clinically justified, patient-centric, and often a combination of these. And this is a real-world example. So what we see is that this is capillary electrophoresis reducing with where the range of the heavy chain and light chain is 97.8 to 99.5 for 14 drug products lots. The mean is 98.3. The tolerance limit is 96.7. And then the proposal was, since there is a limited data set and limited experience in the commercial environment, the acceptance criteria for heavy is more than 95%. The clinical batch ranges is 98.1 to 99.5%. And there is no clinical justification of limit. And this may not be an issue. This may be absolutely fine, but it's not us guessing, it's companies telling us why this is acceptable. So yeah, I think the first bullet point really shows this. On the other hand, we often see that when companies can justify the limits from a clinical perspective, regulators may still ask for tightening of the limits if the batch results are tighter than the actual limits. And this tightening may be prerequisite for approval or requested to be done after a specified number of batches have been produced, even if the limits can be considered to have no negative on safety and efficacy. And what this does is to punish companies which are have a very good control of their production and with a low variability. And so where do we go from there? So here are some personal reflection on this revision of the Q6 guidelines. A lot is still applicable, as I said. Add aspects introduced Q8 to Q12 and 13-14 when they are in force, impacting on setting specs. Update the characterization section and the appendix as appropriate, taking new methodology and product understanding into account. Consider if the scope should be widened to some of the products now excluded, taking added complexity into consideration, that realizing that advanced therapy medicinal products are quite different from monoclonals. I think avoid a too high-level document allowing for considerable regional interpretation, because global harmonization of specification will not be possible during an application process, but more likely to be reached if the principle for selecting attributes to be tested and how to set acceptance criteria are agreed, and therefore it need to go down in quite detail on these aspects. Some further reflection on that: expand the section on justification to clarify principle in more detail and avoid the split message. And we had already in 2011 a workshop with industry and EMA, and the outcome was clinical qualification is considered the most important aspect when setting the acceptance criteria. And this is not only linked to levels in batches used in clinical trials, just to make sure that clinical qualification was used in this wording more of what we now perhaps call clinical justification, or you call it patient-centric. And another aspect is reach a common understanding of how much data is needed to claim a limit to be clinically justify that. How many patients should have been exposed to a certain level? Is it sufficient that one patient once got an aged product with a certain level, or do you need a larger cohort to be able to say that yes, this is a safe and efficacious level? Consistency is important to verify that the process is under control, but that can be handled by other means through the pharmaceutical quality system, and it's through trending and internal action plans. And this is more of an GMP aspect, but make this clear in the application how you are going to verify your consistency. I think it should also include some options beyond the level that how can you go beyond the level seen in the actual clinical trials to justify limits. That is by using prior knowledge from relevant products. You can say, oh, by criticality assignment, this is not a critical attribute, sorry, and therefore I don't need to test it, or I can have wider limits. What are your experience? I think experience from dose finding studies are often very useful, because you can recalculate the levels for that particular commercial strength and say, okay, we have given this patient this amount of impurity and with no problem. Use some in vitro methods and non-clinical data. And I think often we can see from efficacy aspects, we can use such method. Until now, we have seen less of safety aspects, but maybe this is coming as well. And the principle for setting the specification should also be applicable for those biologicals which are not included as such in the scope. So just finalizing with some experience from PRIME, the accelerated and products with unmet clinical need, and COVID-19 products. And as you all know and have experienced yourself, it's a very challenging task. In some cases, we have clearly novel concept mRNA products, adeno 26 products with no or little previous regulatory experience and less prior knowledge to lean on. The justification for specification limits should be for CQA should be linked to clinical performance rather than solely derived from statistical method used at tolerance interval. And as this is the same as discussed above. And the unmet clinical need may allow for a different benefit risk assessment and allowing some uncertainty of safety and efficacy at the outer boundaries of the acceptance criteria, but should still include a clinical justification based on, for example, in vitro studies, prior knowledge, which could be shown to be relevant for the product in question. And in this case, to minimize the potential risk that in this case, it may be reasonable to ask for a revision of the acceptance criteria after a certain number of batches has been produced. There is a major difference in clinical experience of prime products, which could be very rare diseases with few patients treated, and COVID vaccines, where you had 10,000s of people involved in clinical trials and billions of people that were exposed in a short time frame after approval. And therefore, they are not really, you can't really compare them. And I would say that that would also the situation we saw for instability aspects, that for the COVID vaccines, we knew that there were really requests for materials. So even if they had shorter shelf lives initially, everything was used in very short period. While if you have an obscure, very rare disease, and you come with this product, this will not be the case. That then you would need 18 months or 2 years or whatever to have a commercially viable shelf life. So with that, I end my presentation and thank you very much for listening. Thank you.
Many thanks, Matt, for sharing your very informative and valuable personal reflection on the ICH Q6B and the Q5C. We have one question from Dan. So you briefly commented on the predictive stability modeling, including kinetic models and stats analysis to support more limited real-time data. Do you have suggestions? Do you have suggestions for achieving greater acceptance of such modeling approach?
I would say that as I said that the best thing you can do is to approach the agencies with scientific advice and do it early on, just to make sure that and explain your case, what you have seen. I think we see it more and more, and we have, as was discussed last year and this year, we see, particularly for shelf life setting, that we absolutely see this coming, but for companies to be more assured and doing the right thing from the very start, approach the agencies with, for EMA, you can ask for scientific advice, or you can ask for a discussion with the innovation task force or the quality innovation group, and to have some early feedback. But I think that is what we can say today. But it will also be, I guess, with a more spread and a union way of looking at these, that when they are more established, I guess they also will be more readily acceptable, but until now, the one we have seen was, I'm not sure it would have been accepted on its own, but in this case, it was to we had some few batches with full-time stability, and now we could support this with that model, and so that I think is where we are today, but I'm sure we will see it much more in the future. Thank you.
Okay, thank you, Matt. It's time for the next speech. I think we might be better to keep the questions from team and from Dr. Adlan to the maybe discussion session.
Yeah.
Yeah. Okay. So. So our next speaker is Dr. Anne Yelolis. Anne holds a master degree in stats from Oxford University and a PhD in stats from Waterloo, Canada. From 1991 to 93, she was the head of the mathematics and the stats section of Share Research. Then she joined the Information and the Stats Division of NHS Scotland, the ISD, studying as a head and the principal statistician of the Scottish Cancer Therapy Network within ISD. She became assistant director of ISD before establishing Quantix in 2002. Quantix was involved in bioassay in 2008 and has been worked in this field ever since. Evaluation of assay performance is often challenging. Today, we are very pleased to have Anne to speak to this topic. Please help me welcome Anne Yelolis.
Hello, everyone. Can you see my screen? I've just attempted to share my slides.
Yeah, very clear.
Fantastic. Thank you very much. Okay, thank you very much for the invitation to the conference. I've never been to this particular conference before, and I'm really excited to be taking part. It's really a lot of fun. So I've been working in bioassay for a good while now, and involved in trying to redefine how we evaluate assays, how we validate assays. And this is a talk to try and present some actually quite old ideas. These ideas are not new, and I'm not pretending they're mine either. But it's really just a discussion of the different approaches to bioassay performance validation. So I'm going to be speaking about relative potency bioassays, and specifically really for batch release bioassays. And I'm going to start by thinking, what properties do we actually need of a bioassay in order to try and think about things that we're trying to validate? So I'm sure anybody who's been involved in validating has come across this expression before. What we really want is that the result is very likely to be close to the true value, that is the true potency of the batch that we're testing. And the question really revolves around what we mean by all these things in the quotation marks. But having said that, for batch release, what we really want is to make sure we've got a 0-1 decision here at the end of the day. Either the batch passes or it fails. And what we really want to achieve is bad batches failing, or be very likely to fail. And that's what I'm calling sensitivity here. So the assay needs to be sensitive to bad batches, that is to identify them as out of specification. And it also needs to be very good at passing batches that are really good batches. So ones that are really in spec should pass. And I'm calling that specificity. So that's the kind of basis from which all of this is developed. And the attributes that validation guidances ask us to check, amongst others, are accuracy and precision. So first of all, I want to just recap for us what we mean by accuracy and precision. And they're two separate attributes. These graphs show, each graph has a dot in the middle here, in the middle of the line, the bright white dot, which is supposed to be the true value. And then there are a cloud of more hazy points, which are supposed to be the measured values. And if they're clustered closely together, like here or here, then we say they're precise, that is high precision, as compared with over here, where they're widely spaced out. And if they are on average close to the dot, then they have high accuracy. And if they're on average far from the dot, such as here or here, then the accuracy is low. So it's really important to make sure that we understand those two completely different attributes. So the question is, you could have high accuracy, and that would allow you to have slightly less good precision, or vice versa. So it's a kind of two-dimensional question. And what we've been trying to think of is how we could improve this to create a single measure of quality, which addresses the questions that we're trying to make sure we achieve, the attributes that we're trying to achieve, through a single measurement, which would be a combination of accuracy and precision. And we're calling it total error. Now, total error has been defined lots of times. Everybody, I think, probably has got a good idea of what we mean by total error. The first definition that I found by just Googling is total error equals bias plus some multiple of the coefficient of variation. So a really simple formula. But it's not always that. Some people call it total analytical error on the absolute or the proportional scale. And that's specifically defined as the absolute bias plus a multiple of standard deviation. Not quite the same as this one. Or you can have this proportional bias, which has proportional error, which has percentage bias plus some multiple of the coefficient of variation. So we've got various approaches, all of which are very similar. So I'm sure everybody's familiar with at least the ideas of these. This is an illustration which I borrowed from FinBiosoft's website, which is very nice. It illustrates it beautifully. This helps us to interpret what we mean by this total error expression. So let's suppose that the blue shape illustrates the distribution of the measured values for a single lot. So if you repeatedly tested a lot, you would get variable results, and we assume that they're normally distributed. And we'll come to that later. And that normal distribution will have a particular mean, which is denoted by the green line. And that mean won't be the same as the true value in general. The difference between the true value and that mean is called the bias. At the same time, this distribution has a particular spread or a particular precision, and that's assessed by the standard deviation. And if we combine the bias with some multiple of the standard deviation, let's call it 2. So I've said, let's suppose we choose Z as 2. Then we get this quantity here, which is marked on the bottom. Bias plus 2 standard deviations, like that. So this length is the total analytical error, the absolute total analytical error. That's an example. So what does that mean? It tells us how far away from the true result, the true value, the result could be with a certain probability. And of course, it depends on this assumption of normality, as I just said. That's the transformation between the limit that we've calculated and the probability. But all we really know at this point is that big is bad. We don't know how big is too bad. All we know is that the bigger, the worse it is. And there's just a couple of examples here of values of Z which give you those probabilities. So that's the total analytical error. It's a good measure in a sense, but it's hard to interpret on its own. Here's another measure, and I've also borrowed this plot from the FinBiosoft website as well, something called measurement uncertainty. And this is another combination of bias and precision, only it's done on the square scale and then square rooted. And there's 2 different interpretations of that as well. But again, how do we interpret this? What does it mean other than big is bad? It's kind of like the root mean square error, if you're familiar with that, the square distance from the true value. So we're not very much further forward because we've got some numbers, but we don't really know what to do with them, or at least I don't really know what to do with them at this stage. So let's try and improve the interpretation by linking these quantities to specification limits. And when we do that, we can start to look at the probability of a lot being within those limits, given the error, given the bias and the precision. And now we come up with this measure called process capability, which I'm sure lots of people have heard of. I don't know if you know, but this was first proposed in 1988. So as I said, this is not new, of course. These are old ideas, which have been used a lot in industry, in the manufacturing industry. But I think they're not probably used maybe enough in our industry. So the process capability measure incorporates performance against a standard. And by standard, I mean the difference between the upper specification and the lower specification. So this is like the gateposts through which our lot has to pass based on our assay. Some people call it Taguchi Capability Index. And if you think about the elements of this expression, I'm sorry, this is a rather horrible expression, we have a sigma, a standard deviation. And that's kind of very similar to what we saw before, the SD in the previous definitions. And we also have this other number here, mu, which is the average of the measurements, minus T, which stands for the true value of the lot. And this, of course, is the bias, the difference between the average, that's the peak in that blue graph, and the true value. So what we have here, and actually this expression on the bottom as well, the whole thing is just the measurement uncertainty with K set to 6. There was a K in there. So what we have here is something which is very similar to what we had before, but the thing we have on the bottom is like what we had on the top previously. So big is good in this case. The bigger the CPM, which makes sense, it's a positive capability, that's a positive word. We want it to be big. Big is good. Okay, so the question is, how big is good? And in this case, we can come up with something which is a lot more intuitively interpretable, because we can use this expression for a lot that has the true value at the midpoint of the limits. The probability of a lot being out of specification, testing using this assay, is this expression here. It's twice the cumulative normal density function. I hope lots of people have heard of that, but it's something you can look up in tables. You can get it from Excel, etc. And I'll show you what it really means a little bit later. But it's this thing evaluated at -3 times the CPM. So this thing is giving us an approximation to the probability of being out of specification. It's exact when the bias is 0. It's not exact when the bias is not 0. So CPM is quite a nice combination of accuracy and precision. And here's a little table that shows you roughly how values of CPM relate to probability of out of spec. So as the CPM goes up, the probability of being out of spec reduces. And at 1, you've got a sort of a benchmark at 1, which gives you a probability of being out of spec of about 0.3%. So a fairly low probability of being out of spec. So that's process capability. What I want to do next is to apply all these concepts to an example. And the example I've stolen from, or borrowed, I suppose, from USP General Chapter 1033 on biological assay validation. And in that chapter, there's an example. It's actually a simulated example, which is fully worked up in terms of accuracy and precision. So the example is, it's a simulated study, as I said, which has got 5 test samples in it. It's a dilutional linearity study. And those test samples are theoretically derived from the reference standard to have expected or true relative potencies of these 5 numbers to give us a nice linearity measure. So 0.5 up to 2 on a log increasing scale, geometric scale. And each of the samples, in theory, was tested in 8 runs each. And here's the data. These are the results. We've got expected relative potency along here, and you'll notice this is on a log scale. and the measured relative potency up here, also on a log scale. Now, I'm going to do a little bit of slightly messy statistics here. It's brief, but I have to do this because, as I mentioned before, we're working on a log scale for relative potency because we need that for the normal distribution stuff to work. So the measured potency, the X I'm going to call log of the relative potency, and it's a normally distributed quantity. The thing is that typically we report bias and precision on a raw scale, so a percentage on a raw scale, so that's a kind of raw scale measure. But we need to convert those back to the log scale in order to do our calculations of these total error expressions. So what I'm going to tell you just again is the definitions for bioassay of bias and precision. And again, 1033 gives us these definitions. Relative bias is defined as the ratio of measured to expected potency minus 1 times 100%. So it's like a function of the recovery here. Also just going to mention that when I say expected or true, they're interchangeable as far as this presentation is concerned. Now, so we're going to see that the relative bias is reported as this percentage, but the question is, what does that tell us the mean of the distribution of the log relative potency is? Well, on the log scale, the bias would be the difference between the log measured potency and the log expected potency. That would be that distance between the true and the measured mean. And just using this expression, that just tells us that the bias on the log scale can be got from the relative bias from this expression. So don't worry about why that is if you're not really familiar with this stuff. But that's how we're going to get from relative bias to bias on the log scale. Very similarly, we have the expression for intermediate precision is also a percentage on essentially on the raw scale, and it involves the standard deviation of the log relative potencies. That's sigma there, and the expression there is the log, the standard deviation of the log relative potencies. So it's quite easy to get that sigma from the intermediate precision by this expression, which is really similar to the bias one. You'll notice it's log of 1 plus the thing over 100. So it's quite easy to implement. And one more thing I want to just explain is that intermediate precision is fine, but that applies to one run of the assay. In our example, we were going to assume, we have assumed, that the assay format is 3 runs. So it has a smaller variability because it's based on the average of 3. The variance of that average is going to be, the standard deviation of that average is going to be called sigma subscript reportable value. And exactly the same thing applies. That sigma can be got from the format variability as it's defined in the guidance. So I hope that didn't blow your minds too much. It's really just the background to get from bias to mean and from intermediate precision or format variability to standard deviation in order that we can calculate these expressions. So here are some numbers. You'll find most of these in the guidance if you're interested. So in the guidance, we have these 5 levels, as I mentioned, 5 expected values. We have relative bias calculated based on these numbers. And we also have an upper confidence limit for that relative bias, which is the thing that's proposed to be, or upper and lower confidence limits actually, that are proposed to be the things that we compare with our target acceptance criteria. So I've just chosen really rather arbitrarily to use the upper limits as the example. So these are the bias examples I'm using. And similarly, I've chosen the upper confidence limit on the format variability for my variability value. So we can quickly and easily apply these formulas to get to the bias on the log scale. So this is how we calculate those. It's just simply this function here and this function here. So we've gone from the percentages to the means and standard deviations on the log scale, which don't look very intuitive at all, do they? They're not useful things to know about on their own on the log scale, but we'll get back to the raw scale in a minute. So now we can plug the bias that we've calculated and the SD, the standard deviation that we've calculated, into the total error function. And I've chosen Z to be 3. So that gives us a very high probability of the value being at most this thing that we've calculated. So we just plug those numbers into this. We've got so for example, for this one, we've got bias 0.074 plus 3 times 0.065 gives us this. And similarly, all the way across there. Still not very intuitive. What we can do, though, is we can back transform this thing. And that basically gives us the upper limit, the number below which we expect 99.9% of cases to be on the measured over expected scale. So they're all around kind of 1.3. And in the middle here, this is the value below which we expect this ratio to be. That's the highest value we expect it to be. So it's worst case, the measured is 30% higher than the expected. That's what we're saying there. But we still don't have a feeling for whether that's good or bad for the assay because we haven't compared it with anything performance related. So now let's move on to CPM and apply that to this as well. So here's the function for CPM, just to remind you. What I haven't yet defined is the limits. So in the example, we have set the limits at the lower limit is 0.7 for relative potency. The upper limit is 1.43. And because we're working on the log scale, we have to transform these to the log scale. So we've done all that and we've calculated from this that CPM is, and I've just kept to the middle point here because it really doesn't make sense to have true values that are kind of outside the limits. So we're just staying with the value of 1. We're assuming that the true value is 1 or 100% here for this example. And it turns out that CPM is 1.026 under these assumptions. So the assumptions that go into this value are bias 10.1%, format variability 6.7%, and the limits. Plug all those into the function and we get this. It's above 1, so that's a good sign. And the probability from that expression that I showed you comes out at 0.208%. So that's saying that the probability of a randomly selected lot whose true value is 1, the probability that the test result using this assay for that lot, the probability it will fall out of spec is about 0.2%. And bearing in mind that the upper limit for the spec is 1.4, that's fairly consistent with this value, the 1.34, which says that 99% of values will be at most 1.34. So we've got a very small percentage of being out of spec when we're at the middle. So that's all quite nice. But what bothers me as a statistician is that the probability of being out of spec, which is the thing I really like, is an approximation using the CPM. And we can very easily just do it properly. So let's do it properly to start with. Because that probability of out of spec is what really helps us to evaluate the sensitivity and specificity of the assay. Sensitivity, just to remind you again how I'm defining this, when the true value is out of spec, that means outside 0.7 to 1.43 in our example. That means if the true value is outside that, you really want to reject that batch. Certainly from the regulators or the patient's perspective, you don't want batches that are out of spec getting through into use. And also there's a risk there with clinical trials, of course. If you've got a super potent lot, you might get safety issues. If you've got a subpotent lot, you might get efficacy issues. So there's all sorts of reasons why we don't want to accept out of spec lots. And on the other side of the coin, we do not want to reject a lot when it is not out of spec. And that I've called the specificity. And obviously from a company perspective, you certainly don't want to waste material. So these are the two aspects of that, which I think is important to get right. So we'll move through this and just show how we're going to do this. So reminding ourselves that the relative potency is log normally distributed. We've seen this before. The actual values of the value for the mean, if we think about this, the log of the measured potency minus the log of expected potency, which I'm going to call mu true or target, is the bias. So that tells us that the mean of the log RP is actually the true value plus the bias. So this is where we can start evaluating bias and the variance. So let's take the example where the true relative potency is 1, which gives us a mu t is 0 because we're logging these things. The relative bias is 10%. That gives us, from that expression I showed you before, beta is 0.095, and intermediate precision 10% gives us sigma is 0.095 as well. It's the same transformation. So this graph shows us the distribution of the measured relative potency for such a lot. That means if we repeated the measurement lots and lots of times and drew a histogram, it would look like this. And from this, if we put the specification limits on 70 to 143 or 0.7 to 143, we can now calculate the probability of being out of specification. What it is it's the amount of the proportion of cases which are either above the upper specification or below the lower specification. So it's the area under this curve. And you can see that it's absolutely minute at the bottom because we've got a positive bias here. So you're very, very, very unlikely to fail on the lower end, but you've got a reasonable probability of failing at the higher end there. And we can just calculate this using normal distribution theory. And it's this cumulative normal density function again that I mentioned before, just like we saw in the CPM calculation. And it's just like this. I'm not going to bother you with those details. We can look these up in a table. So let's now add a row to the table that we had before. So previously we had the probability of out of spec being this value, but that value only really applies when the true value is 1. So now, not only can we calculate the correct value here in the middle, but we can also calculate the probability of being out of spec for all of these other values, which is, I think, is a useful thing. What it tells us is really good, actually. It tells us that when the true value is 0.5, you've got 100% chance of being out of spec. That is, well, nearly 100% chance of being out of spec. You're very, very unlikely to pass with this assay if your true potency is 0.5. Similarly, when your true potency is 2, you've got a very, very small chance of passing. So that's good. It's very sensitive at those values. In the middle, when the true value is 1, which is in spec, we really want that one to pass. The probability of passing is very high. So I don't know why I've miscalculated that. It should be 1 minus 0.003, which is not 99.992, is 99.997, I think. So this is looking good. The other thing we can do with this formula is, as well as doing the calculations at each of these points, we can also expand that to any true relative potency value we like, using the same criteria, the same properties. And what we can now do is we can say, okay, what values of the true potency give us a chance of being out of spec that's at least 90%? So this is sensitivity. And what we can see here is that if the true potency is as low as 0.59 or as high as 1.41, then you've got a chance of at least 90% of being out of spec. And similarly, we could draw a line across at 10% and find where it cuts the line. And this shows us that if you're between 0.69 and 1.2, you've got at most a 10% chance of being out of spec. And of course, you could draw that line across at any value you liked, 0.1, 0.01, etc., to find out what values of true potency are likely to get a rejection. So I think this is quite a useful interpretation. Now, so far we've just talked about specific known true potencies of lots. And for manufacturing, of course, what you really want to do is look at the distribution of the manufacturing process and work out what proportion of lots randomly chosen from that process are likely to be rejected. And that's a slightly different question, although it's really similar in terms of how we work it out. So let's suppose that the manufacturing process, the log of the true values, again, we're going to assume it's normal distribution with a mean mu big M and a sigma squared product. I don't know why I didn't call that mu product, but mu and sigma squared of the process. And these are completely separate and independent from the assay, of course. But with these values, we can say that the distribution of assay results from randomly selected lots from that manufacturing process, not a lot with a given potency, but a randomly selected lot, has this distribution. So it's just like we saw before, but it's now mu m, the process, that's replaced the true value we had before, plus the bias. And then we're going to add the variance of the process, that sigma squared product, to that variance that we had before. So we've basically changed the mean a bit, and we've added variance to the variability because of the variability between lots. And then what we can do, and this is getting slightly complicated, so apologies for that. What I've done here is to take all of those parameters as fixed. We've got the limits as before. I've assumed that the variability of the process is about 8%, so it's slightly bigger than the variability of the that we had before. And I've taken a couple of, or 3 different sets of relative bias and format variability values and combined them with all of these. And then we get these curves here. And you can use, so it's just similar to what we looked at before, but it's for the process instead of specific values. And now you can look to see what your chances of being out of spec are. Now we're basing it on the mean of the manufacturing process. And of course, with relative bias of 0, which is the third example, the dotted one, the curve is symmetrical about 1 because the manufacturing process is actually centered at 1, which is in the middle of the spec. Okay, so that's manufacturing processes. Now, the last thing I want to talk about is interval approaches to the evaluation of the qualities of the assay. And again, this is quite an old reference as well, so you can see how we're kind of behind the curve on this. Boulanger and colleagues published a paper in 2007, which was titled Risk Management for Analytical Methods Based on the Total Error Concept. And they were trying to reconcile the 2 aspects of validation in the same way as we're trying to do here. And they came up with something called a beta expectation tolerance interval. And that was defined as an interval which is expected to contain 100 beta% of bias values, that is distances from the true value. Okay, now this is very similar to a p% prediction interval, but usually I think, I'm happy to discuss this one, usually prediction intervals are based on the actual observations rather than the bias, because we're actually looking at where we expect to see the results fall. So they're very, very similar, and it depends what you mean by what you're predicting, actually, what the interval is that you're trying to predict. So you could interpret this expectation tolerance interval as being 100 beta% prediction interval for the bias. So if this observation was the bias, then it would be exactly the same thing. So just an aside there, that this is what that is. It's a prediction interval in terms of calculations. I've borrowed this graph from the Boulanger paper, and it's very similar to the first graph that we looked at. But what's important here is that we're looking at the bias. This is a x minus mu true, the true mean, and x is the measured value. And notice that these are normal distributions again. And what they proposed doing was calculating this interval, the beta expectation interval, and declaring the assay valid if that interval falls within predefined limits. So this ties really nicely in with that definition of a good assay, which is that the result is very likely, that is beta likely, so beta needs to be high, very likely to be close, that is within minus lambda to plus lambda, to the true value, because we want this difference to be 0, really. And as it moves away from 0, the result moves away from the true value. So we're going to calculate this beta expectation interval based on the bias, and we're going to look at where it sits in relation to our predefined limits. Here's the definition of the interval. So as I said, it looks just like a confidence interval or a prediction interval. It's the calculated value of the observations, the mean of the observations, the distance of that from the true mean, and then we're going to add and subtract some multiple, which is, you'll have seen this before, I'm not going to explain this in great detail, the information's here. It's a multiplier times the estimate of the standard deviation of the x's. And what's really interesting about this, I think, is that this looks exactly like the total analytical error, really. It's the upper, total analytical error is kind of the upper limit of this, with a particular choice of z. If we chose this value to be z, then this will be the same thing as the upper limit of that interval. So again, there's not a lot new, it's just the way we're interpreting it that's different. So what I've done here is to calculate those limits, those beta expectation tolerance limits, for the example from the 1033 chapter example. And we need to do it on a log scale because we're relying on normality. And I've chosen beta to be 0.9. So that means we've got a 90% or expectation tolerance interval here for the reportable value. And this time, instead, I've used these values for the relative bias and the format variability. These aren't the upper limits, these are the reported ones, because that ties in more with what Boulanger and colleagues were saying. So you'll see that the upper limit does this for the 5 different targets or true values. The lower limit does this, and I've just plotted on there the 0 line, which is what we're aspiring to. We want to be here. And now I've simply back transformed this axis so that what we're seeing here, instead of a sort of funny arbitrary number, which we don't really understand, here we've got something which relates to the ratio of the observed value to the true value. And of course, now 1 is the ideal. Okay, so this is the same plot as before, but I've just shifted this axis onto the log scale. Sorry, back to the raw scale, from the log scale. And now, Boulanger and colleagues suggest that a 15% is a reasonable width for lambda. And back on the raw scale, that translates into 1.15, and then the symmetrical end on the other end, 1 over 1.15. And remember that this is for individual values. So this is we're talking about a tolerance interval, a 90% tolerance interval for these values, and we want that tolerance interval to fall within these limits. So what we have here is that at each of the 4 lower true potencies, we have success because the interval, that value there in the middle is just within the interval. We've got a pass for the first 4, but at 2, a relative potency of 2 or 200%, we have a fail because the interval goes outside the 15%, saying that some values will be further than 1.15 from this limit, or the interval, the 90% interval includes values which will be above that limit. So the interpretation of that would be that the assay is not valid at the top. So the assay is valid if the interval falls within the predefined limits. So I think that's quite a nice thing. And I'll just go back to this for a second, because based on the variance on the intermediate precision and bias, actually just based on the bias, only this one failed and all of the rest of them passed. So it's a nice single summary of that. And the final thing I just wanted to mention is that the thing I really like about this paper, which I haven't illustrated here because it's kind of repeating what we've looked at before, is that the other way of looking at this is completely symmetrical, really. But the other way of looking at this is, instead of calculating the interval, you can actually calculate the probability of falling within these predefined limits, just like we did before. But here we're talking about the biases rather than the specifications and the actual values, so it's a bit different. But the methodology is the same. So we can calculate that probability of falling within the predefined limits, and then your assay would be valid if that probability was at least beta. It's exactly the same thing, it's just the other way around. And the refinement that Boulanger and colleagues add to this is that they calculate a lower confidence limit for that probability. So it takes account of the fact that you've only got so much information here. So instead of just reporting that calculated probability, you have a lower confidence limit for that. And that's in tune with all of the guidances, the USP guidances currently, which work with confidence limits on things. So all of these are leading to the same conclusion, that if these things hold, then the result is very likely, likelihood determined by beta, to be close, closeness determined by the limits, which were those lambdas that we saw, to the true value. So that's really nice. I like that. The final thing I just wanted to tell you about very quickly was that this beta prediction interval for the actual reported value. And here we can just take exactly the same formula we had before, but get rid of the bias and just report the actual observed value. What this then allows us to do is give us intervals at each predicted value, 90% prediction intervals around the result. So now we've got this scale, which is relative reportable value on the raw scale. And now we can put the specifications on there. And what we can now say is that, like we were saying before, at the top end, when the true value is 2, we are almost 100, well over 100%, not well over 100%, very nearly 100% probability of being out of spec. Same at the bottom, well in spec in the middle. So it's just a different way of looking at it. And this is a 90% prediction interval. So in summary, we can combine accuracy and precision into total error in a lot of different ways. I particularly like that last way of doing it, and we can get a single interval, or we can do it as a probability. And of course, an interval's got 2 ends, so maybe a probability is the better thing because it's just one number. And it gives a simple assessment of combined performance. As soon as we add the upper specification and lower specification, that informs the diagnostic accuracy, that is the specificity and sensitivity. And we can add manufacturing performance, manufacturing process parameters, which gives us same information, but it's now about the process instead of the individual values. So that's a summary of what I had to say, and I look forward to discussing it with you. Thanks.
Many thanks, Anne, for sharing this very practically meaningful and useful stats approach for IC performance evaluation. I really appreciate it. For this session, we're a little bit running out of time, and we're supposed to take 15 minutes break. And I think if you have a question for Matt and Anne, we could you could bring up in the discussion session later on. And we're going to take another, I think, maybe 10 minutes break. We start at 11:28 for the panel breakout sessions. So our last speaker, but not the least, is Paul Faya. Paul is the Director of Development Statistics at the Eli Lilly and Company. Supports CMC research and development activities for diverse portfolios of biological molecules. He has expertise in analytical development, process development, and stability modeling. Paul is currently the chair of ASADIA non-clinical Bayesian industry working group. He holds a PhD in statistical science and an MBA from Baylor University. We are pleased to have Paul to give us an update from the CMC Bayesian Scientific Working Group. Podium is yours, Paul.
Thanks, Jim. Can you hear me?
Very clearly.
Okay, great. I'm starting to share my screen, so let me know when you can see it. Can you see this, Yoon, the screen?
Yes, that's fine.
Yes.
Okay, great. All right, thanks for the introduction, and thanks for inviting me and the committee to this conference. I'm glad to be back. Last year I presented some discussions on regulatory concerns and issues, and so today I just wanted to give kind of an update on what we've been up to, some opportunities, and just kind of what we're seeing in the regulatory landscape that is encouraging from our point of view. So yeah, that's the outline and update from our group, regulatory developments and future opportunities. So this is currently the membership of our group. It's grown in the last year or so, and which is encouraging. We have 13 members right now, primarily all from industry. One, we have John Seaman, who's from Baylor University, so one academic. And I'm actually, yeah, Yoon, I'm trying to get him to join from Health Canada. It would be really great to have someone from a regulatory body as part of this working group. We used to have someone from the FDA, but that person dropped out. So we're hoping that we can pull someone in from agencies such as Health Canada. But it's been a really good dynamic group to work with. And I also want to say for anyone on this call, if you're interested or have someone within your groups that are interested in contributing or being involved in this work, feel free to email me and let me know. So what have we been up to this year? We've had a couple of major deliverables. The first one was a paper that is currently under review with the journal. And this was born out of a lot of things we were seeing in the literature, not only from industry, but also conference presentations and so forth from FDA statisticians about using doing stability analysis using mixed models, and in particular using tolerance intervals based on those mixed models to set shelf life. And so there were some things that were concerning to us about that approach, and we also felt like it would be very important to provide a Bayesian analog to that, which we feel is kind of more intuitive and a little bit more elegant to get to. And so anyway, this was a good preview for it. We're hoping it'll be published sometime next year. But essentially, it's a two-part paper. First half kind of provides a critique of the literature that's come out recently on tolerance intervals using mixed models, and then the second part is a Bayesian counterpart. And we give a couple of examples, one using a non-informative prior, and then one using, say, a prior based on prior experience with that molecule. So look forward to that coming out soon. And then the other major deliverable that we had, which is actually coming out this December, is we've been working on this for the last 2 years, is a book on case studies for Bayesian methods in CMC. This kind of the genesis of this was well, when we started in 2019, this working group, we did an industry survey and we wanted to know, what are the needs, what are the barriers to adoption of Bayesian methods, in CMC. And there was an overwhelming response of we need more examples, worked examples with code and everything to help us, get started in this area. And so out of that, we started working on this idea. And so we finally finished it this year. And I think it's a really great book. I mean, first of all, I don't get any royalties for this, so I'm not trying to promote it for my own game, but I think it's a really great book. It provides a really wide variety of articles from a variety of authors, but also some that are very introductory. So someone who's new to Bayesian can get their hands on there and implement it using the code. And then there's some case studies that are a little bit more advanced. But I think in general, it provides really good kind of inspiration and motivation for the application of Bayesian methods in CMC. And there's also an accompanying website. So if you're interested in trying some of the examples, you can just download the code instead of having to type it out yourself. So anyway, that's this December, both the e-copy and the hard copy should come out. I think you can pre-order on Amazon now. So that was a huge accomplishment for our working group. I would say that most of the members contributed a chapter or multiple chapters. So what are we doing, or what are our plans for next year and the future? One thing that we really want to do is to draft an industry guidance on Bayesian methods for CMC. And we don't know what venue necessarily it'll be published in, but our goal is to really just give high level advice and best practices. I know Jose, who spoke a couple of days ago, was talking about it'd be great if we had maybe some, guidance around what priors to use for certain situations and so forth. And so I think that's the goal of this thing, is to not be prescriptive necessarily, but in the spirit of typical guidances, just give kind of boundaries and ways to approach, typical CMC scenarios using a Bayesian method. And I really hope we can get good regulatory input or even co-authorship on this type of thing, because I think it would go a long way. And again, if you're interested in being involved in that, this is one of our big deliverables for next year. We also feel like it'd be good to provide some commentary on recent ICH drafts and revisions, which I feel like are very encouraging and kind of Bayesian oriented. And so we might do something like that. Again, I don't know in what venue, but I think it's important as an industry to make commentary. And then finally, as a promotion, just NCB conference is coming up. This is a biannual non-stats conference. It's happening in Rutgers University this year. And so we'll be fairly involved in that conference. I'm on the organizing committee, and so really encourage you to attend if you can. There'll be some really good talks and courses as well. So we'll have some involvement there. So that's kind of what we've been up to in our future work. So I wanted to spend the rest of the talk just going over some things that we're seeing that are encouraging. This is a totally non-technical talk. It's really just kind of a overview of ideas and topics that are coming out in the regulatory space. And Tim, I wasn't in your breakout session, so if I'm covering stuff that you guys already discussed, especially related to analytical methods, I apologize. But this EMA Prime Toolbox, so Matt's touched upon this in his presentation earlier this morning. So Prime stands for Priority Medicines. And I really love this toolbox. It's essentially a voluntary scheme, right? It's supposed to provide enhanced support for development of medicines that target unmet medical needs. So it encourages things like early dialogue, optimization, and speeding up of evaluation process and ways to overcome challenges. And this, I feel like this toolbox is great. It's very, in my opinion, like lined up with Bayesian thinking. It focuses heavily on prior knowledge, and it defines prior knowledge as knowledge from development and manufacturing experience, not only based on the current product, but also based on similar compounds and products and processes. And it also refers to even scientific and technical publications as a source of prior knowledge. So the toolbox argues that by using prior knowledge, you can not only shift the time point, right, of certain stability, process validations, justification of specs, i.e. get it done faster, but also you can even make them redundant or not necessary. But then there's also this focus on platform knowledge, which I think is coming, this kind of wording is coming out in a lot of different places, not just with EMA, but basically from similar formulation, manufacturing processes, analytical tests, and so on. And I think, one of the challenges for the industry as a whole will be to, and I think this question came up in a previous presentation, is how do you define similar enough, right? Matt's talked about, I think, structural similarity, but I think that's something that as we delve into this prior knowledge world, we'll have to really figure out how to define that. So the toolbox covers a lot of things, and I just wanted to highlight 3 that I thought were interesting. It talks about comparability studies, so using prior knowledge to predict the impact of scientific manufacturing changes, and also even the choice of quality attributes to be studied. And in terms of setting specifications, again, using platform knowledge to determine the impact of potential CQAs on the development program, and a comparison and justification that it basically argues that you have to actually compare and justify any differences between the existing product and the one that you're using prior knowledge for. And then also in stability, right? Saying that stability models, again, like Matt's mentioned, can be generated from prior knowledge. And there it does say structurally similar molecules. And one question I had here, and I think Tim asked this as well, is why can't this toolbox apply to the development of any medicines, right? Why should it be limited to the prime medicines, right, that are dealing with unmet needs? I would suggest that these kinds of tools should be available to any medicine, right? If we can justify similarity of products and justify what prior knowledge we're using, that should be relevant regardless of what the priority of the medicine itself. This reflection paper, another EMA paper, came out in 2021. But again, this one, Rick Burdick, I think, mentioned in his presentation a couple of days ago on reflections on statistical methodologies for comparative assessments. And this was kind of, to me, encouraging and also a little discouraging. Encouraging in a sense that I think it's one of the first times I've seen, a regulatory kind of publication coming out actually outright stating Bayesian methods. In this case, they talked about Bayesian credible intervals, and discussed what it is and how potentially they could be used for comparative assessments. But there's a statement here that says the acceptability of Bayesian approaches to inform regulatory decision-making remains controversial. I don't know why it's controversial. I think it's more that it's perhaps not well understood, but I'm not sure why that would be controversial. And then it also goes on to say, acceptability of Bayesian approaches would require persuasive operating characteristics, but also an acceptance of conceptually different interpretation of data analysis in general, which it makes it sound like that's a bad thing or hard thing. I think that's probably a better thing, right? I think Catherine touched upon this in her presentation, how the interpretation is actually much more intuitive and aligned with how scientists think about probability and intervals. So anyway, so this I think was good, but also kind of a little discouraging to kind of get a sense of what do regulators think about Bayesian approaches. It sounds like it may still be controversial, I guess, in their minds. And the last thing I wanted to touch on were the revisions to Q2 and Q14. And again, Tim, I wasn't in your breakout, so you may have talked about this already, but I was really encouraged by these two revisions when I read them. So were a lot of people in our working group. We felt like they were completely turning Bayesian. Neither of them actually mention Bayesian methods, but the language and what is being proposed is completely in line with how Bayesians think and do analysis. The definition of a validation study is now an evaluation of prior knowledge data or experiments, right, to determine the suitability of an analytical procedure, meaning that prior knowledge may be enough to say that the method is validated. And in fact, they give this really cool figure here in the draft guidance saying you have the validation objectives, you go into your validation strategy, which is either an evaluation of existing data or the actual experimentation, right? So it's one or the other. And so you basically evaluate the new experiments or the existing data and write document a final validation conclusion. So it's a very different paradigm of how we look at validation, no longer just a standalone study, right? But it could, the justification of the suitability of method, the source of that evidence can come from a lot of different places. And they outright say that, right, in the actual document itself. Suitable data derived from development studies can be used in lieu of validation data. And also, again, also this reference to platform analytical procedures can lead to abbreviated validation testing if scientifically justified. And I think this is just repeating the same thing. Data gathered during development studies and suitability tests even can be applied to validation, right? So that's another good source of suitability, system suitability tests, right? Give us a good source of method performance. And so that can be used to support understanding of validation. So Anne talked a lot about earlier this morning, total error, and I'm not gonna go over that, but this is emphasized as well a lot in ICHQ2 R2, which was very encouraging to see. What I didn't see, and I wish I had, was a reference to something like a posterior predictive distribution, right? They talked about how accuracy and precision can be evaluated simultaneously using a prediction interval. And so they talk about, or a tolerance interval. I hope they can add to this list a posterior predictive distribution. They do say other approaches may be acceptable if justified. So again, this is all encouraging, and I hope that there can be more explicit references to Bayesian approaches in the future in this work. And actually, I wanted to highlight a couple papers that came out in 2021 and 2022, which are essentially completely in line with this new revision of ICHQ2. So Steve Novick and some of his colleagues from AstraZeneca published a Bayesian statistical approach to continuous qualification of a bioassay. And then earlier this year, our working group published a follow-up paper on continuous method validation beyond one-time studies to characterize analytical methods, which is exactly what ICHQ2 is saying, right? Validation is not a one-time study, and our paper basically discussed how can you use, development data, how can you use, say, robustness data or GMP results, right, to combine it all into one global understanding of method performance to characterize the method. And in the paper, we talked about downweighting historical data, right, relative to the validation study, but it sounds like from the trajectory of Q2 that that's not even necessary, right? If you have enough evidence, quantified evidence that the method is capable or suitable for its intended purpose, you don't even need that validation study anymore. But all that to say, we've been thinking and writing about this for a while, so it was nice to see that ICHQ2R2 aligns nicely with the way we're thinking about validation. And so for ICHQ14, this is more of the global development kind of strategy. It's a new document. And again, they make reference to platform analytical procedures, saying that development can be abbreviated in certain tests, in certain cases totally omitted, right? So this is very similar language to the EMA paper on the prime toolbox, where you can abbreviate or eliminate certain tests based on historical data. And they say in general, data gathered during development studies can be used. So again, reiterating the same thing that I've been mentioning. So this is all super encouraging to me personally and to our working group. I know that these ICH documents are currently under review. All the comments have come in, so it'll be interesting to see the final products. And let me see what is, okay, so one last thing I didn't want to mention is there's a big discussion on knowledge management in ICHQ14. And again, the definition of prior knowledge to me is exactly aligned with that EMA interpretation, right? Internal knowledge from a company's development program as well as external knowledge, right, from scientific publications. So it seems like that this idea is kind of being consistently proposed across regulatory bodies. And again, the idea of using existing platform analytical procedures is part of knowledge management. And both of these, whether you're quantifying platform knowledge in some way or quantifying prior knowledge, are all very easily done through Bayesian methods. The last thing I wanted to point out is even in the industry itself, I don't know how many people are familiar with these conferences by CAS, but the CMC Strategy Forum and WCBP coming up early next year. So this is not a statistical, these aren't statistical conferences, right? They're industry conferences, but there's a roundtable scheduled for this year on using Bayesian statistics and stability modeling and leveraging prior knowledge. And this idea of leveraging prior knowledge has been discussed in the past in WCBP events. And to my knowledge, this is the first time where there's an outright topic, right, about Bayesian statistics, which I don't know who proposed this, but I'm encouraged that it's out there. So it's nice to see not only are things coming out from regulatory bodies, but also even from scientists, right, scientific community here, talking about the need for Bayesian methods. So anyway, I just wanted to conclude by saying that we're encouraged by what's happening in kind of the regulatory communication. It feels like there's progress happening. It seems that the idea of prior knowledge and platform understanding are really being embraced by industry and regulators. And we really feel like there's opportunities for industry regulatory collaboration. I know that Jose has talked about how they've collaborated with Health Canada and UNE, and I hope we can do more of that, not only with Health Canada, but other regulatory authorities. I feel like Health Canada is setting a really good example for others to follow. And then, as was mentioned again earlier this morning as well, ICH Q1 and Q5 are under review or going through some working groups. And I think, again, there's big opportunities there to evolve those documents to include some Bayesian ideas. So anyway, this was a fairly short presentation, but I think I just wanted to give some food for thought as we go into the discussion section of this session of what's happening and the opportunities for the future. So I think that's all I had, June.
Many thanks, Paul, for the updates. Glad to know you guys have made such progress on different directions. And thanks for the comments and on the relevant ICH quality guidelines, and also some suggestions to better communicate and working together with regulatory agencies. Now we have about 4 minutes question time for Paul. And I may read from, there are some questions in chat. So there's one comment, I believe, from Jose. It's quite long.
Yeah, it's
Do you want to just ask it, Jose?
No. You were talking about controversial and the word that appeared in the EMA.
Yes.
Reflection paper. And back in 2013, Professor Efron, it was the 250th anniversary of the Bayes theorem, and he used that word. I mean, that's from the abstract of a paper that he wrote, and he talked about the suspicion of statisticians still after 250 years. So
yeah, it's unfortunate.
Yeah, it is.
It's unfortunate.
Yeah. So, I mean, all he's saying that it's still after 250 years. Yeah, it is still controversial.
Yeah.
Okay. Let me see. It's no questions, just maybe comments. It's comments from Dan. This was an excellent summary of evolving regulatory perspective. From Janet, agree with Dan. Thank you, Paul. No questions. Any questions? Okay, if you guys have questions, we could keep it to the discussion session. Now there should be the discussion session should be started within 5 minutes at 1:22 p.m. and it will be facilitated by Christiana. Should we have a break, or should we continue directly to the discussion, Christiana? What do you think?
We are basically at time, so I would kind of start, because 5 minutes it's maybe too short if everyone agrees. Yes.
Okay. Yeah. Okay.
Right. Thanks. Okay. So thanks, June, for the excellent sharing, and welcome to the panel discussion for session 3. First of all, I would like to really thank again all the speakers for the great presentation, and I would like to invite them to join the panel by switching on their camera, if possible. And I welcome June again, who has kindly agreed to be part of the panel discussion as well. So a long day for you, June. Thanks for this. All right, so do we have everyone? Yes. Anna, yes, you're there. Okay, good. All right, so what we will do is to use this time to ensure that we can address any residual questions for the speakers, as June was mentioning, and of course trying to continue systematic reflection on various CMC topics that we have covered, like specification, stability, analytical strategy, process developments. And, you know, I see that all these topics are kind of synergic in a way, right? Because we have talked about specifications, but also consistency, control strategy. And so just to open up the panel, I would like to ask all the panelists if they want to comment on connection and interdependencies among the topics that we have presented during the session, just to kind of warm up a little bit, and then we can go into the detailed elements from the audience. And please use the chat if you want to participate or raise your hand for the audience. So I don't know who would like to start. Maybe Mats, you would like to go first with some reflections on this, because you had the broader view in your slides.
Yeah. Now, I think what we do see here, and I think what should be considered is it will be, and particularly some of the presentation were really into statistics, and I think this to get success from applications, I think it's really important to realize that these people are not as, we don't have the deep knowledge as others have. And the problem with many of the, and I would say most of the regulatory agencies is the problem that we do have statisticians, but they are more into the clinics and the clinical trials and so on. They are not CMC statisticians. And what we have learned from discussing with is that they don't really, sometimes don't realize the difference we have in sample sizes, that we are not talking about 4,000 people in a clinical study. We're talking about a much more limited amount of data, and how to make stability predictions out of that. And I think that is having said that, I think this is clearly something we need to really look for, and to have people, but they are not that, it's not that many out there. So the problem is to get them to us, and and maybe they are not full-time statisticians for this, but having this kind of competence is really needed here. And so where we can go, I hope we can get there, and we can, I would say perhaps that is something for EMA to look for, that having a broader, having some personnel which are more specialized on that, and can take it into account. And yeah, maybe I stop there with the first one to see whether. Thank you.
Thanks, Mats. And I think that the initiatives that you mentioned, like the innovation group, the quality innovation group, you know, could also be ways
Indeed.
for engaging EMA, for instance, in this kind of new thinking,
Yeah.
right? Maybe not
Indeed.
too product specific, but in a broader way, right?
Yeah, no model sort of interpretation of models and so on, and what to expect will clearly come there. So you're absolutely correct. They will have the idea with this group. I don't know if you know it, but all the people know it. But it's a small group with like 8 people from QWP, BWP, and inspectors working, but having a lot of experts surrounding them with different competencies to use whenever questions come up. So any question you will have on, as we did discuss earlier, that anything you will have on models and so on will be a good way of going to them and then having feedback.
Thank you, Mats. Anyone else wants to comment on what Mats said, or other ideas for, let's say, bringing more connection among these apparently separated elements of regulatory framework versus operational and statistical? Yeah, Anne.
Yeah, hi. I've got a couple of comments actually. I think there's a bit of a thread here with the Bayesian discussion. I know that in clinical stats, and Matt's mentioned that all the statisticians in the regulatory area in Europe tend to be clinical. And in the clinical area, there's been a big resistance to Bayesian, the use of Bayesian stats. It's coming, but it was certainly really slow. And I think, that's something that maybe this area is lagging behind a little bit, but I guess it'll come eventually. And the other thing I was going to mention was that Paul mentioned the sort of suspicion of Bayesian methods regarding total error and the prediction intervals, and I absolutely agree with that. I think I was going to mention it, but then I realised I didn't really have time to say what all I wanted anyway. But I think that definitely there's a role in, as obviously is already happening, in making the case for Bayesian applications and the use of Bayesian methods in this area. So those are my thoughts. Thanks.
Yeah, thanks very much. I see Tim.
Yeah, I guess Anne and I are on the same planet, because she said exactly what I was going to say. I was specifically reacting to Matt saying that there's limited data in CMC, and that's not true at all. There's plenty of data that can be harvested using Bayesian methods, and I believe that through the last 2 conferences, the statistical community has agreed that that's the path forward, is to break some barriers, both the statistical barriers and moving away from traditional frequentist methods to Bayesian, but also some of the technical barriers, such as the lack of appreciation of platforms. I posted a question about the fact that we know a whole lot more than we pretend to know when we develop a product, and we don't really use that information effectively. And I think if we want to facilitate development for the future, I have no doubt, and I won't be here and I can't implement it anyway, that Bayesian is going to be the replacement means of facilitating CMC development for the future. Even the products are moving in that direction with, for instance, mRNA vaccines, which are ripe for platform thinking. And we now need to change the way we do technical and regulatory business to adapt to those or to innovate in those spaces.
Yeah. And the team, does it mean that we could also push for having more explicit reference to Bayesian statistics in the guidelines? There is a comment, for instance, on the presentation by Paul, which mentioned that it could be good, for instance, in ICHQ2 to have more Bayesian specific wording to really foster the implementation of this approach. Would that help?
It would more than help. I always believe in pull rather than push. We're pushing Bayesian now. And guidelines could pull for it if the people who wrote guidelines were aware of its power to do many things, including probably reduce costs of medicines, because the development costs would be reduced by, sharing information across projects and so forth. So, the pull of guidelines has been my bread and butter. I mean, I had to help write some guidelines to get that to happen. So again, I'm afraid that, as much as I like these conferences and discussions about it, informing is not as powerful as doing. And again, I don't think we're going to be doing things, consistently until there's the pull from regulators or from guidelines to do so.
Indeed. Thanks, Tim. So we have another couple of hands raised. So I see Illert and then Julia. Yeah. You are muted, sorry. Yes. It's still not really, we cannot really hear you. Yeah.
I hope now it's better.
Yes.
Yeah, perfect.
Perfect.
Yes,
Thank
much better.
you. Yeah.
Thank you. Yeah, my name is Tobias, Tobias Eilert. I just put in my last name. Thank you.
Okay.
Yeah, I wanted to agree with Tim completely. As he said, there's so much information which we do not kind of harvest. And there I was also thinking about prior elicitation. That means not only taking data, which is everywhere, which we can use, but we can also get from talking to the practitioners, to the SMEs, because to be honest, their information, their feeling about methods and their processes is usually pretty precise. They're really good. I think there we should also get better and better to measure these, to put them also into distributions and use them. I think there is still like a huge dimension, huge universe to kind of detect and put into information. So I think there's, for Bayesian methods, still a huge step to do. Also, I wanted to throw in, especially for when we have problems coming in, troubleshootings, for example, we also think about using Bayesian networks, like causal networks, to use this to make deductions from statistics more acceptable, addressable for the SMEs. I think this is very far away to go. I don't think that will come soon, but I think there also Bayesian can help us a lot to make statistics more understandable.
Thank you. That's great. Thanks very much, Thomas. Julia, do you want to go next?
Sure. So I'm going to be the controversial or contrary person here. Is I never understand why we work so hard to push a statistical class of methods or philosophy, if you want to call it that, instead of pushing that we have new ways to solve problems that may be, I mean, very much like unmet medical needs. I'm very intrigued about the potential for applying Bayesian approaches on stability, because I feel like the current, traditional ICH and COVA model isn't serving us well when we have too much data. And the random effects model, I think, doesn't serve us well if we don't have volumes of data. So I'm certainly open to applying Bayesian methods where we don't have current great solutions. But I feel like, somebody just mentioned that using a Bayesian approach could make the results of the statistical modeling more accessible or more understandable by the scientists we work with and reviewers. But I find it difficult to get past the labeling of things as Bayesian or not Bayesian. And I consider myself a pragmatist to the nth degree, where I can go back more than 10 years, where I pulled on using chemometric multivariate methods and machine learning in its early days in combination with classical statistical methods, because we had a huge problem to solve, and it took a combination of all those things to solve it. So when I hear that we just want people to use and accept Bayesian methods, the wall kind of goes up for me. And I wonder if it does for other people too, because it's a one last thought on that is at Moderna, certainly we have combined data across multiple products in a platform, but we don't formally call that Bayesian. And it hasn't been necessary. I mean, for example, process characterization experiments for control strategy of vaccine variant products can certainly build on the data from the original strain process characterization, but it's not necessary for us to go Bayesian to do that. So I guess I'll kind of throw it out there that I think by emphasizing the statistical method instead of the solving of a problem, I think we may be actually slowing down the adoption of Bayesian as a new class of useful methods.
So you mean, sorry, trying to translate it as now everyone knows as a non-statistical expert. So focusing on the problem solution and, not on the methodology, which can be applicable to many other aspects. Yeah.
Right. And yes, exactly. And I think that by focusing on the methodology, we may be reinforcing the perception that this is something brand new and different. Instead of, several people have said these techniques have been around a long time. This is not brand new. And why are they controversial? But I think we're almost framing it that way, that here's some brand new way to do things that is different. And, I'm not sure why we are doing that. So just my
thank you.
My engineer turned statistician point of view.
Yeah, I think that someone wants to react. I've seen first June raising his hand, and then I think in order Laura and Tim, and then maybe we can switch topic because we have many things we want to discuss. June,
okay,
you want to?
I think Julia brought up a very good point. Like we should focus on the solution. There could be any solution, but patients could be one of them. Very powerful, very useful one in some situation. For example, for the stability analysis, patient could play a role, but there might be other approach. And if we could, I don't know, there could be a publication to compare the patient and the other approaches, both use of the prior knowledge to solve the stability analysis for a biosimilar product, for example. That could be a approach it's one way to promote the any solutions could solve a problem, and the patient might be a better one compared to the other one. So we need a comparison to compare the patient approach to the other approach. There are two approaches, both of them using the prior knowledge. The second is we may start from small. For example, we know in one field, for example, using stability as an example again, there might not be well-established conventional approach to solve some problem. For example, when we only have a limited information for the new product. In that case, we may draft a guideline specifically for that, stability analysis, the patient's approach application in stability analysis, stability study, specifically for that. Because I saw FDA guidelines, some of them occasionally only have one page specifically for one situation, for one product. So there could be we could focus on some guidance specifically for a small area, be very focused on one thing. The third is there could be the industry people could think about publish, the guidance before, like they got 100% support from regulatory agencies. Because I saw the publications is about how to guidance on the patients reporting for the academic publication they publish on, as I remember, on Nature. So you published it, you could more or less you could make the influence, create some influence. Because first I remember when we got to the first submission, patient submission in quality CMC review, we could not find the relevant guidelines, so we searched on website of, we found that guideline. That guideline is for the academic publication, patient statistics, but it's still useful for us. So there are three things. One thing is like we should compare the patient's method to another available method to try to show the advantages, if there could be some obvious advantages for the patient's method. Second is we focus on something small, specific. For example, stability of study. The third is we published what we think is a good guidance on some journal with a good impact. Thank you.
Thanks, Joe. Right, so let's go with Laura in order of, I think she was, yeah. Hi, Laura.
Hi. I just wanted to say thank you to Julia for pointing that out. I do think that focusing on the problem, on how we solve the problem is important, and that Bayesian methods can be an important tool in our toolbox. I noted when Anne was talking, I really enjoyed her presentation. I noted she did have a Bayesian approach in there, but she never said Bayesian. She was talking about how to solve the problem. And that was really refreshing to me. So I wanted to say thank you to you for that, Anne. And I'll stop talking because I see other hands raised. So thanks, guys.
Thanks very much. Tim, you go next.
Yeah, I think I'm going to temper the contrarian's remarks by saying the promotion of Bayesian was not that Bayesian solves everything. It was that Bayesian is invisible right now, and that we need something, that pull, to make it part of the toolbox. So again, no, I'm not going to use Bayesian to calculate an average, but I am going to wish that I had it in my toolbox and felt that there was no risk in my using it if I felt it was the best approach to deal with a particular problem. So that's the way it needs to be thought about. I think that's what Paul is advocating for, is that we at least get it out there as an opportunity and do whatever's necessary to put it into full implementation or production when it is useful.
Thanks, Tim. And maybe, Catherine, if you don't mind, I would like to give the word to Paul, because I think he will react to something that Tim has just said, and then we will go to you, Catherine. So,
yeah,
Paul, you want...
I'll just say something very briefly. Julia and Laura, no one disagrees with you. I agree with you. I think all of us agree that it's a tool in the toolbox, right? And we encounter a problem and we find the best tool to solve it, whether it's Bayesian, frequentist, whatever. But you can't deny the fact that frequentist thinking and principles are ingrained right into what we've historically done and the way that regulators look at things, right? Ideas of confidence intervals, tolerance intervals, p-values, they all know that. It shows up in guidance documents, right? Calculate a confidence interval, show me the p-value. So it's more of getting the water levels to the same kind of point, maybe, as opposed to like just pushing a methodology. It's more about getting familiarity with regulators. So I think Matt's was talking about the clinical side, right? Or it was Anne, where they've made advances, right? They've had to go through the same pain. They've had to do exactly what we're doing to get to the point where now they can do Bayesian adaptive designs for clinical trials, right? So it's become embraced in the regulatory circles in the clinical world. So it's more of an education, promotion, movement. It's not saying Bayesian, 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 right? This is the way to do it. So I think we have to keep that in mind. Is like, we live in a frequentist mindset. Everyone thinks that way, confidence interval p-values. And we're just trying to even the playing field a little, not saying that it's it's the way. And even in our paper, we called it a probabilistic solution, right? We didn't say a Bayesian solution to stability, we call it probabilistic. So we understand that. And I think, yeah, that's the effort. And I think that's all I wanted to say there.
Thanks very much. Catherine, now finally to you. Thanks.
Hi, yeah. So I think it's a, it's a worthwhile point that, we talk about what addresses the problem best. And in fact, that's what those of us who use Bayesian methods have been doing. I think when we argue for why we use them. And so I would just make a counter argument that why then is there pushback immediately to the word Bayesian, right? If it's just another tool in the toolbox and we're focusing on what methodology best addresses the problems, then we should fight against, frankly, knee-jerk reaction, right? I also think that it's not useful to focus on comparing results. I think Rick made the point very well on Monday. I can't remember exactly the way he said it. I actually wrote it down somewhere, but I can't find it quickly enough. Comparing the results from one methodology to the results of some other methodology is not a way to validate the second methodology, right? That's not, what does that tell us? We know that when we use diffuse or non-informative priors for many ways of framing the question, we're going to get the same answer, but the Bayesian has different interpretation, which can be useful and more intuitive sometimes. We also know that if we use informative priors, we're not going to get the same answer. So we're asking a silly question. I don't really understand what the value is in saying we need things that compare the two methodologies. We know that it's another tool in the toolbox. Sometimes it's going to give a similar answer with different interpretation, and sometimes it's going to get a different answer for very good reasons that we need to think about when that is a better way to address the problem.
Thank you. Thanks, Catherine. Yeah, very good point. Anyone wants to have share any reflection on what was just mentioned? I think it was quite clear and relevant. Right. So I think that we can try to move back to the broader set of topics that we discussed. There are, maybe we can start from a couple of general questions, and then maybe we have to address also some other questions more specific from the audience to make sure that everything can be clarified while we are all together. So one point that was for Mats really, and we quickly touched on it, but maybe we need to expand, is the question on prime principles, right? Scientifically and empirically based, do they apply to programs other than accelerated programs? And if not, what is the expected addition? And probably, Mats, this is again for you to expand and comment. Thank you.
Yeah, I think if you read when you read the toolbox document, it's quite obvious that certain parts of it is really to a specific situation where we urgently need something. As I was mentioning, the concurrent validation. Normally we would expect 3 PPQ batches or something like that. If we do have a situation where this is really urgent, we have some indication this will be good, still we are willing to take the risk to go with the concurrent validation instead of waiting for the normal 3 batches. On the other hand, I would say that there are, if you read the document, there are things which are applicable now. It is after the consultation phase, there was an update. Initially, it was just prime and breakthrough therapy. It is now widened to say that it may also be applicable for other with unmet clinical need, but not with the stamp of a breakthrough or prime. I would go maybe a little bit even further to say that if you have good reasons, the stability model based on prior knowledge could be applicable for more standard applications as well. And I think there are also on the small molecule side, there are some of them which is not very controversial in that sense. So it's, I think what we do see there is going to be more, and some of it may be applicable for others, but I would say that everything isn't yet, at least. And again, that if you are going to use it for something else, please check with the agency before you start, because otherwise you may end up in throwing away a lot of time for you have to redo something. So it's urgent it's really important to get and discuss with us whether is this doable, and if you have a good reason, we may say yes, it is.
Yeah. And you know, I think that one point of attention, really practically speaking, Mats, is that under prime designation, by default, we normally have more interactions with authorities, really to check, right? Because, you know, to check where we are going and have really also strategic discussions on risk-based approaches in a non-prime or not yet the designated products. You know, this is where probably it's difficult to identify the right channel for interacting. You know, can it be through specific let's say meetings like, you know, scientific advice or similar, or, you know, is it you know, through maybe, as we already said, non-product specific discussions, at least for the introduction of new technologies or new statistical approaches. But, you know, this is where probably sometimes it's difficult for the companies to orient themselves and to invest time for that. You see, practically speaking. You are right. Yeah.
I clearly see, but I think what we, coming back a little bit to the links I posted on the chat, where a lot of those has been the effect of the interested parties meeting between BWP and industry, that we saw that there is a need for more on prior knowledge, which I think we talking about GC here, but we are also a lot on and I see Tim's comment here that of course you can build a lot of your new processes can be built on prior knowledge with similar molecules, because we are not saying you should have the absolute optimal process. You should have a reproducible, well-controlled process which delivers the quality you have qualified in your trials or by other means, and do it over and over again. And that is the background. So therefore, if you look at an RDNA or monoclonal process, 90% of them do have the same purification process, because it works, and it's the difference is so small. And I guess we have seen similar things for mRNA vaccines, where with the new strains, that you didn't have to repeat everything, because you could rely on what was done with the initial strain. So I think this will come more and more, I'm sure.
Yeah, I think this will come more and more also with respect to non-clinical models, maybe, right, to support the identification of critical quality attributes, and also with respect to bridging with clinical testing that we were discussing yesterday. On day one, right, with during the keynote. Thanks, Mats. Okay, so I would like to go to the other questions from the chat, just to make sure we can cover them. We had a couple of questions for Laurent Straudiot. So the first one actually was, I think, addressed directly during the discussion. There was another one that I would like to share, So is the following: are you able to discuss what online monitoring are you doing with your spectral sensors, and which components of the soup are you mapping, and have you developed calibrations for them? So can you please comment or try to address if you can, Laurent?
Yes. What we do actually, and I'm sorry I will not be able to give all the details because we don't have already publication to describe everything. But what I can tell you actually is that we have different concentration and different mixtures of the lipids that we use for liposomes. We've seen now we are using DOPC cholesterol, and we have also the MPL and QSNT1 in the mixture. And so we have different composition concentrations, and we take the spectra of the different samples, and from there we create the chemometric models to have calibration curves, if you want, with different concentrations. And maybe one thing that we can tell, as you noticed, that we have the SNIR in water solution. So all good chemists will tell you that it's not possible to use SNIR in water because you would have lots of interference the water on your spectrum analysis. So here we use specific probes in a specific way to avoid the water interference using a diffractions method. So that's how we do actually. I hope it answered the questions.
Thanks, Laurent. We can check with the audience if it's clear with the person who made the question. I cannot retrieve it now, but hopefully
we can send me a direct message if needed.
Yeah, indeed. Thanks, Laurent. And you know, as we have you here, I remember that you actually all had some open questions, right? That are really related to, let's say, the continuous processing, digital twin, and things like that. I think one that is particularly interesting is related to specification and stability expectations in this continuous processing setting, which at the end of the day is also related to the issue that sometimes we have with having more acceptance of parametric release approaches for biological products. And you know, what I think you were trying to stimulate that, but maybe you can expand a little bit on this, is any more considerations on these in guidances, because probably Q13 doesn't go too much into the detail of that, considering that specification and stability guidelines are being updated. Can you elaborate a little bit on these elements of stability for incoming materials and so on, that for which you would think there is a need for more framework?
Yeah, so you have seen in the described process, we have two incoming materials, so two stocks actually, one organic and one aqueous. So the organic is a mixture of isopropanol, ethanol containing DOPC, cholesterol, and MPL. So the process runs, it runs for maybe a couple of hours, I would say. So we need to demonstrate that for the durations of the process, the quality of the solution is always the same, and that the last fraction that will be stuck into the process is the same quality as the very first fraction. So we have a continuous process with identical quality, and so for what we have done so far is we took samples, we measured the concentration, and we checked that the concentrations were always within specification during the storage, I mean the in use storage, I would say. We've done similar things for the other hands of the process. So when we have generated the liposomes, so you still have about, between 20 and 30%, 35% of alcohol in the suspension of liposomes. So we have also we needed to also to monitor or to make sure that it's stable for the duration of the operations, but also for the next steps. where we have to start for the dye filtrations to remove the organic waste. So what we have done, we have taken a kind of worst case scenario, or where we store this product for 2 days, and we monitor the stability for over 2 days, knowing that we'll never go for 2 days of stability, but as we demonstrated after 2 days, the concentrations of the liposome were the same, the compositions and also the size distribution were identical, and yeah, I think that's what we did actually. So is it good enough?
Understood. So it's more like a risk-based approach, and you assume that this is gonna be a condition that will be valid also in case of a future manufacturing or process changes. And you would like to see more information and guidance on these elements, right? On you know, as a feedback from the. Thank you. So it's clear, and I don't know if anyone wants to comment on this, but I think it's a valid point. All right, thanks very much, Laurent. So I don't know if there are other questions from the speakers. I personally have still a couple, but maybe I want to make sure that everyone else.
Yeah, Matt wants to answer or give us some comments.
Mats.
Yeah. No, I think what you show here is a really good case of worst case whole time that taken into consideration whatever may happen, and if you can show that, I think that shouldn't be difficult to what we normally would see as far as I can understand it here, that what we normally would see for an whole time during in a process validations. So and again that taking it, realizing that this will never ever, we will never come as, we will never have it for this period, but if it's stable, excellent, then we know it gives you a freedom if something happens, that you do have that validated and clear. So I think that would be a good, it's a good way of showing it.
Thanks.
Thank you. But I have also maybe a follow-up questions for you, Matt. So you have seen that in the compositions we have MPL, we have TOPC, and so on and so on. So we have different lots. So do we need to regularly validate these holding times in terms of stability, or is it one good for all?
I would say that you should have incoming specifications for your material that would allow you to draw the conclusion that this will work as exactly as the earlier batches did. So I think maybe here what we do see sometimes is fermentation media where you may have some high variability and lesser character stability, where we could have problems, but for this one, it's, if you have good specifications for the incoming material, that should be sufficient.
Thank you.
Good, thanks. So just before I continue to talk, want to make sure that there are no further comments on the topic and on what Laurent has presented. Okay, so if not, I actually would like to ask a question to Anne. I was really intrigued by the examples of total error that you made, and you showed really different options and verification opportunities for this, because for me, many people are still struggling with unifying a definition of total error, right? As you clearly stated, and so I wanted to ask you an expert feedback on what you have seen in ICH Q14, and if the ICH Q14 definition, which of course doesn't enter too much into the detail, with maybe something in the annexes, but not so much, if this is something that is aligned with what has been your thinking and the concept, the principles that you had behind your presentation, or if there are opportunities for clarification in that space, especially on the ATP and the total error.
Yeah, I have to admit I'm not overly familiar with ICH Q14, and I'd have to check it out and see how it fitted in with all of this. So I can't answer that one clearly, Christiana. I'm sorry about that. I'm not really involved in that sort of level of analysis. I'm much more kind of more at the front end, I think. Yeah, sorry.
No worries. It was really my curiosity, because a lot of principles that you have, I think, fit very well with the principles of Q14. Yeah. And I think many people in the Q14 teams from industry were also connected to USP. Yeah. So I was just curious to have your opinion on that.
That's it. I mean, I think I'm much more aligned with the sort of biologic specific guidances. I think that's really the difference. But it's a really good point that we need to align with those too, because I'm sure lots of these ideas have been more developed in the analytical field.
Yes, indeed.
And I think that maybe the biological area, Tim can comment on this much better than I can, I think. But I think it's really good to align those guidances, because they're very aligned in many ways. But in other ways, the biologics approach maybe needs to be a bit different. But I think probably in this case, it doesn't. It's a principle.
Yeah.
But the relative thing and the log scale and all of that, I think is quite specific to biologics, possibly.
Yeah, there is the reason why I was asking your advice and reflection is that in the Q14 there is one of the current examples. I don't know if it will remain. It's in the published draft that is on a potency test as well. So that's why I was making the connection with what you have presented, but it doesn't go at all into the level of detail of potential statistical approaches like normally ICH do, because they don't want to be too constraining, considering that they last for many years. So if it's too prescriptive, it's a problem. But I was just curious.
No, I think that's the same thing with USP, but I will have a look at that. Thanks for the heads up on that one. I'll make sure I check it out. Thank you.
Good. All right. Anyone wants to comment? I know, Tim, you have been very much into the topic, so I would like to put you on the spot and have your opinion on that, if you wish, because you were also connected with the analytical.
Boy, I'm so glad that I don't have to offer my opinion. You actually asked for it. I always feel like I'm interfering in the discussion. I don't remember. I mean, you know that I was personally involved with you and Phil Borman and others, and as Q14 was evolving, and I can't say that I remember exactly either what ended up in there. I don't think, however, there's any ambiguity about what total error means, and for that matter, I hope there's no resistance against it, in the sense that going back to maybe, to paraphrase the discussion about Bayesian, it makes sense. It answers a question. I think it answers it more effectively when you revert to fitness for use or analytical target profile than does the old way of doing things. And I do hope that, again, similar to Bayesian, we modernize the way we think about things. So the whole idea, whether it gets in a guideline or not, is all a matter of trying to modernize the way we think about doing things, and maybe better than in modernizing, improving the way we do things so that we can serve patients better. I mean, it all comes down to, it's not a matter of, Bayesian this or total error that. It's about, doing good pharmaceutical development and control to serve patients. So, in my mind, and my motivation is always to pick up things and do them, improve matters, especially when, there's always the fear of risking failing validation. And we had a discussion in the analytical group that Rick Burdick brought up, which had to do with, well, what if you get contradictory results between the individual parameters basis of validating an assay and the total error basis? And my answer would be the total error basis is the answer. The individual parameters basis of it was a patch. It was an approximation to what we really wanted to know. So let's, change our question from, showing that a relative accuracy passes or fails, and then separate from that, that a intermediate precision passes and fail. They don't operate separately.
Mm-hmm.
So again, I don't know, I think I'm rambling in a sense, but at the same time, I hope I'm helping you with the how do we transition into this, and to whatever extent Q14 has offered it up, like we would like, other guidelines to offer up Bayesian. I think we need to have it out there to play around with a bit to get more comfortable with it.
Yeah, no, indeed, Tim, thanks. And I can kind of reassure you and Anne that luckily, as USP 1220 was published more or less in parallel to the draft of Q14, we tried really to keep consistency in content and in the current annex also some work that we had done with Phil Borman team is also cross-referenced at the moment, right? So for as an example of a total error. So at the moment we are trying to keep consistency, but I think what Anne has presented is much broader, because it has an application to more deep on biological tests which have specific challenges, and so it was illuminating. It brought a different perspective on the topic. So
now you want to hear a quick irony?
Yeah.
Bruno Boulanger and I almost came to blows over total error. I was so against it at one time because I was so wedded to the individual parameters approach that we almost had the Third World War. So take this as an example of that people can change their minds when they see the value in something, even an opinionated person like me.
It's good to know. It's good. So, for me, just to link this to another topic that we discussed, is, I think that for me, at least in my daily job, the most difficult piece is not only in the right statistical approach for total error, on which I really rely on my esteemed colleagues who are experts in statistics, but there is also what is the input for the total error, the patient-linked input. So basically, how do we define a total error that makes sense, that is linked to, let's say, what we want to see in terms of performance? And the problem I see at the moment, especially in some modalities, is that specifications are, which would be, of course, the natural input, are linked to a technology. And while the analytical target profile, which includes the total error, basically should be technology agnostic. And so I think, it's we have to think about linking what we have seen in the patient-centric approach and the elements that Mats has presented, and this new way of thinking analytical development, more focus on the problem, again, on what we are measuring instead of how, as a connection between the two topics, right? Because it's, if we go for a patient-centric, we have a range, expected range that makes sense on a clinical point of view, on a patient point of view. We have a basis for acceptance criteria for a total error, right? For an acceptable range for a total error. And that is at the moment missing, because the specification guidance has is dated, as Mats mentioned, and has a number of limitations. And we absolutely need to make sure that we make the link to Q14 as we progress with Q6 revision. And I don't know if it's making sense to everyone. I would like to be challenged on this thinking, but it's a practical problem, because before finding the right statistical approach, we have to make sure that we are doing something that makes sense in the context that we are working, right? And that is where I think we are not really there yet in terms of regulatory guidance.
Well, if I can add to that, Christiana, and here I am now offering my opinion instead of being asked for it. I said also, and I reacted when you said there's synergies among things like stability specifications, analytical methods, and so forth and all. There's more than synergies, there's a hierarchy. Specifications, we got to get specifications right before we do anything in the other spaces effectively. Because again, all of the process development and uses of different statistical methods and total error and so forth all need to be aligned with developing towards and ensuring quality, which is defined by specification. So, if I had, since I got Matt's on the on this particular, conference, I cannot, as a statistician, appeal more strongly to the global authorities to get specifications right finally, because as a statistician, I have been hampered to do good work because of the misuse of manufacturing data to set specifications. Yeah. We've been painting ourselves into corners. We can't design studies because we're designing to what we see rather than to what we need. And again, if we can get that question answered, I think we'll have different statistics conferences in the future than what we've had in the past, and we're continuing to have right now.
Yeah. Thanks, Tim. I like the concept of a hierarchy indeed, because there are synergies, but also there is a priority, right? So the input for the subsequent one. This is what you mean by hierarchy, right? So one is input versus others, and that makes sense. Yes.
Well, it's key to the question, which is the beginning of any statistical inquiry.
Yeah, indeed.
Is it good or not?
Yeah, correct. And I think that just reflecting on and expanding on expectations for ATP, and then maybe we can do a final check or round of comments. I think that this predefined expectation for methods can also help a lot with the statistical modeling for stability, for instance, right? Because analytical variability can also create can have an impact on the goodness of the prediction. I think, right, in my limited experience, and taking into account also that element in the expectation, in the performance expectation of a method, depending on the context, could also help. So I'm just thinking, again, another dimension of hierarchy, to use your wording, Tim. I think it's just another thought. Okay, so we have, I think, like 4 minutes, right, to close. So I would like to ask every speaker to make any final remark if they would like to, any impression from this section. Just don't say it was interesting, I liked it. So maybe just try to share what you have learned, or what are points of reflection from each of you, if you don't mind, quickly, and then we can close the panel and go to the final remarks. So who wants to start? Mats.
Yeah, I can start. No, I think it has been a really interesting discussion, both in the breakout we had and in the presentations, and not just because I'm a co-chair of the specs the specs meeting in Basel, but I think that will be a great opportunity to further discuss this on how to use these methods in setting specifications that really tells you that that is directed to what is safe and efficacious rather than what is consistently produced.
Thanks, Mats. Who wants to go next? Paul.
Yeah, this has been really elucidating. I think I would like to make one last point from my perspective. I think there's maybe a fallacy out there that sometimes Bayesian is just another tool in the toolbox. I don't think that's necessarily true. I think any one of us can pick up a calculator or Excel and make a confidence interval. A scientist can do that, a regulator can do that. But probably 90% or more of us could not calculate a credible interval or build a prior. Neither could scientists nor could regulators. So I think to say that it's just another tool is a little misleading. I think it's a tool that's developed in the last 15 years and requires a lot of training and education among scientists, statisticians, regulators. And so anyway, just to promote that idea, that we have to be careful not to think about it as just, oh, let me just pull that toolbox. It's a simulation-based approach that is very different from what you're used to. And I really encourage people to learn it so it becomes a tool that they can use.
Thanks very much, Paul. A wonderful comment. Anne, do you want to go next?
I feel I've already said what I've got from this, actually, Christiana. I think the links between the different viewpoints on all of this is really, really useful and really interesting. And I suppose Tim and I can take some of this away and try and merge it into the USP 1033 thing, and maybe we can consider how we can make it a bit stronger. The total error thing I'm talking about here, in terms of trying to make it, if we can get it in there, then maybe people will start using it more, because I think people are very nervous about doing things that aren't in the guidances, as we discussed in the breakout. Yeah, so that's my takeaway, I think.
Thanks a lot, Laurent.
Yes, I was interested to hear the perspective of regulatory and statisticians, and also I think that for as we are developing more and more new technologies, it's important to make a good correlation and good connections with the different experts, and not wait until the end, but already participate and have regular meeting with the different experts to make sure that we are on the good path and we're not reinventing the wheels or completely deviating from what would be acceptable at the end when we will be presenting a new manufacturing process for a new vaccines product. So that's what I get from these sessions.
Wow, thanks a lot, Laurent. So John, do you want to have a quick final remark before I close the session?
I just want to say thank you each speaker, and each of you actually brought us lots of information, and it's very educational. And I think each of us also benefit from your speech and presentations. So please continue the communication, especially about the patient's statistics. And that's all I want to say. Thank you.
Thanks a lot, June, and I also echo your comments on really to all the speakers. Thanks. It was a great session. I really enjoyed and learned a lot as well. And it's now time to move to the final remarks, reviewing day 3 and previewing the day 4 agenda. Thanks very much.
All right, thank you. Thank you all. Special thanks to June and Christiana for a very engaging session today. Certainly had great discussion. Thanks to Mats and Laurent and Paul for their presentations. Obviously very timely topics that led to a lot of questions and a very great panel discussion. Also wanted to thank Julia and Tim for leading the breakout sessions. There was a lot of great discussion in each one of those, and wanting to have you all just be on the lookout for another survey about today's session. We're very interested in getting your thoughts along the way as we turn our attention to planning for next year. So looking for your thoughts, especially in the free form comment boxes. So if you would take maybe an extra moment as you think about today and form your thoughts for what could we be doing better? What topics would you like to hear more about? That sort of thing. We're going to gather again tomorrow. Tomorrow will be the last session of the workshop. We'll have a very similar format today, and we'll be focusing on CMC statistical methodology, so very applicable studies that we'll learn about, along with parallel breakout sessions once again, one being on the topic of Bayesian, so that may be quite what's the word I'm looking for? Well, it'll be engaging. And the second topic on statistical software for non-statisticians. So please join us again tomorrow at 10 a.m. Thank you all for today, and please be safe. Thank you.